Also, I'm a little bleary-eyed this morning because I've been watching CNN since about 4 this morning with all the updates from Boston. Some major developments in that whole thing this morning. So, but I'm still here. I'm ready to go. Okay. So today we'll get into finishing up with Genesis, female reproductive system, follicle development, menstrual cycle, female hormones, questions, fertilization, cleavage reactions. Towards the end of the lecture, through the next lecture, we kind of morph between human reproduction, stem cells, and development. So all these topics are very interrelated. So you'll kind of see that as particularly like going from fertilization to blastocysts. That's sort of classic human development. So we'll get into that as we get to that. Okay. So just a quick review from Wednesday. In male spermatogenesis, there's a series of cells that are produced that have differences in chromosome number and DNA. Spermatogonia, primary spermatocytes, secondary spermatocytes, spermatid, spermatozoa, and these are due to going from some of the primary spermatocytes. There's a DNA duplication, and following that, you have the meiotic divisions, and therefore you have the loss of the chromosome number and DNA number, leading to the first true haploid, spermatid. And I realize the term haploid refers to chromosomes, but I consider spermatid a true haploid because of the DNA is exactly half of what should be there. And the other thing to remember is from every one spermatogonia, you get 4 spermatozoa. Okay, that'll be very different. The corollary in the female reproductive system is quite different. Then we talked about the structure of the spermatozoa, and we talked about that they're produced in the seminiferous tubercles, transported to the epididymis, then the vas deferens, then the ejaculatory duct. We have several glands that feed in various enzymes, sugars, and so forth. You don't need to know what each one of those do, but in total, what they provide. Then the urethra, and then the urinary tract. Just over again, just one point I forgot to mention on the mechanism of erections. It's a nerve impulse that produces nitric oxide, which activates a guanylate cyclase, so it's hormone-like how it works. GTP gets converted to cyclic GMP. And the part I forgot to mention is the muscle relaxation is smooth muscle relaxation. It's not skeletal muscle. And then there's arterial dilation, also referred to as vasodilation. And then to break down to cyclic GMP, there's an enzyme called phosphodiesterase, and Viagra inhibits that particular enzyme, so it maintains higher levels of cyclic GMP, and therefore more efficient smooth muscle relaxation and arterial dilation. Okay? Everybody clear on that? And then the negative feedback system in males. Remember, negative feedback means constant, consistency. You want this to always be occurring. And hypothalamus produces GnRH, goes to receptors in the anterior pituitary, produces LH, FSH. LH binds to receptors in the Leydig cells, converts cholesterol to testosterone. Also, FSH goes to Sertoli cells, testosterone to dihydrotestosterone. In terms of negative feedback, you make a lot of testosterone, goes into the system. If it gets too high, it turns down, dials down LH and FSH, dials down GnRH, slowly brings down testosterone. When the levels get too low, none of this negative part of it doesn't occur. You bring up GnRH, bring up LH and FSH, make more testosterone, and the cycle continues. So you maintain consistency within a window of concentration. Like biologically, physiologically, there's no really such thing as absolute straight values of consistency. It's always within a window. So testosterone is going like this. It's too low, you make more. Too high, you make less. The idea of negative feedback is to keep it constant. Okay? Note what each cell makes here, because there's some corollaries with the female reproductive system. Any questions on that part that we talked about? The other thing, based on some questions I've been getting from you guys, know the difference between enzymes, receptors, and substrates. If you look at what I have drawn on the board here, what is the substrate for guanylate cyclase? Someone yell it out. Substrate. GTP. What's the product? Cyclic GMP. What is the substrate for phosphodiesterase? Right, cyclic GMP, right? So cyclic GMP is the product of guanylate cyclase, but yet the substrate for phosphodiesterase. Everybody must see this. You can't understand how this stuff works until you know the substrates versus the products versus the actual hormones and receptors. Okay? Questions on that? All right. Then we got into oogenesis. Females are born with about 1 million primary oocytes, and you'll see where that comes from in a second. There's about 400,000 at puberty. There's long resting periods on equal cytokinesis we'll talk about today. There's a potential ovum nucleus produced every 28 days. It's important to realize in males you get a real gamete, a separate cell that's a haploid gamete. Females, you get a gamete nucleus, not a real gamete in terms of DNA, and you'll see why. And finally, we ended with in the follicle and the ovary, there's the oogenic cells. The theca cells convert cholesterol to testosterone in females. Note that in males, remember I have this, Leydig cells produce cholesterol to testosterone. It is essentially the same reaction activated by the same hormone as you'll see, LH, luteinizing hormone. Granulosa cells convert testosterone to estrogen through a very important enzyme called aromatase. Okay? And aromatase, by the way, is this enzyme that's thought in some herbicides to be activated. And therefore, there's all these problems, reproductive problems in frogs and other species because of that. In addition, it's a target of new wave breast cancer treatments because it lowers estrogen levels, and it's a good drug for estrogen-responsive breast cancers and estrogen-responsive ovarian cancers, too. Okay? Questions? So let's pull this together now and complete the oogenesis part of this. And I'll do this. I don't want to leave that there, so we'll go back to this board. As I said, females are born with all their eggs. And it's a generic term we'll use. They're actually primary oocytes. So it starts out in female development in a group of cells called primordial stem cells, just the name that's given. And these guys make a bunch of cells called oogonia, which are 46 chromosomes, 2 cDNA, true diploid. The oogonia then undergo the DNA duplication, and you get primary oocytes. Very analogous to primary spermatocytes. These guys have 46 chromosomes and 4 cDNA. This is what the typical females are born with. So the oogenic lineage gets down to the type of cell that undergoes the DNA duplication. Okay? And in puberty, this is what also is there, primary oocytes. Then at one point in time that we'll talk about today, there's what's called an LH surge in the menstrual cycle, and you get ovulation. And we'll talk a few more details. And this kicks on the meiotic division, but you have unequal cytokinesis, which is the splitting of the cytoplasm. So what you get out of this is a secondary oocyte, which is 23 chromosomes and 2 cDNA. And you get out of it, this has almost all the cytoplasm. You also get out of it something called a polar body. This particular one is 23 chromosomes, 2 cDNA. And this is basically just the nucleus without virtually no cytoplasm. You get unequal cytokinesis. It is not viable. Except in the movie, ever seen the movie Twins with Arnold Schwarzenegger and Danny DeVito? Okay? I don't know if you guys remember that or not. But Arnold Schwarzenegger was a secondary oocyte, and Danny DeVito was the polar body that formed. Okay, that was sort of the premise of that particular movie. Okay? Or one of the premises. Okay? So this is happening at ovulation. This secondary oocyte becomes a potential ovonucleus. This becomes an ovonucleus only after fertilization. And in this case, that the nucleus of the sperm enters into this secondary oocyte. I'll put this board down in a second here. And you get, again, unequal cytokinesis and so forth. So you get another polar body, which is 23 chromosome C DNA. Okay, so this polar body is different than the other polar body. And you get a cell that has an ovum, you get an ovum nucleus. Plus you also have the sperm nucleus inside there. Okay? So the reason you never get an ovum gamete, an intact cell that's haploid, is because you only get this final meiotic division until after fertilization. So the sperm nucleus is inside. Then you get, as part of this process, as we'll talk a little more about, you get the second meiotic division. So at no time, particularly, do you ever have a gamete cell with only the ovum nucleus. You have a cell that has both the sperm and the ovum nucleus. As we'll talk about a little later, the nuclear membranes break down, the DNA recombines, nuclear membranes form, and you get the zygote out of this. So if you look at the total DNA, what's the DNA of the ovum nucleus? 23 chromosome C DNA. What's the DNA of the spermatozoa nucleus? 23 chromosome C DNA. They come together, you've got diploid levels of DNA, 46 and 2C DNA. Does everybody see that? So the reason a female only has a potential ovum nucleus every 28 days is you only see it after fertilization. And you don't have that many fertilizations in a lifetime. Okay? Any questions on that? We're going to get some more detail, but it's important to see what's going on here. Also note, from every one oogonia, how many potential ovum nuclei do you get? 1. From every potential spermatogonia, how many spermatozoa do you get? 4. Okay, that's a big difference between males and females. Okay? And also note the two different polar bodies have two different types of DNA, two different amounts of DNA. They're not the same. Okay? Questions? All right, so now we're going to take this now to ovum development. Follicle development, excuse me. Do this over here. You have to see this prior to the menstrual cycle. So what happens in the ovary, you have a whole bunch of follicles which contain the primary oocytes. And at the beginning of the menstrual cycle, many of these start developing simultaneously. So there's estimated between 20 and 30 follicles start the maturation process. But generally, one wins out. So many start, one wins out. And you end up, this is how blow up it is, you get this primary oocyte kind of surrounded by granulosa cells. It starts developing inside. So this will be the winner right here. This is the guy that starts developing. So 30 of or so develop simultaneously. But generally, one wins out. Sometimes more than one wins out. And that could lead to twins, for example. Okay? We'll get to twinning. There's the next topic on Monday after that. So one wins out. What happens then is, a combination of estrogen, luteinizing hormone, and follicle-stimulating hormone, there's a lot of proliferation of the cells. This is really, really important because it's a critical part of the menstrual cycle. And you'll see what that means hormonally in a little bit. What happens is, you then get this primary oocyte with a very defined membrane around it. This membrane is called the zona pellucida. That's the membrane around the primary oocyte. Then outside of that is a layer of granulosa cells, many cells because of the proliferation. And outside of that is a layer of theca cells. So the theca cells convert cholesterol into testosterone, moves the testosterone into the granulosa cells, converts it to estrogen, which gets into the primary oocyte, which all involves maturation, development, and proliferation. Okay? So that one follicle that wins out really starts growing, really starts maturing. Then you've got fluid starts building up. And we'll just draw this. This is at the edge of the ovary, near the surface. So here's the primary oocyte with all the rest of the goodies. And some fluid starts building up in a compartment called the antrum, which has fluid. Now, one of the reasons for the fluid is, think of a balloon. If you keep pumping in water into a balloon, my kids have done this many, many times. Keep going in, keep going in. At some point in time, that balloon will be very taut, right? Very tight. Any little change will what happen to the balloon? It'll burst, right? So what this fluid does, it provides pressure so that some little change can easily kick out the primary oocyte upon ovulation. So then what happens is, I'll put this over here, I guess. What happens? There's a surge in LH, which kicks on, it's not clear what the exact order is, but kicks on the meiotic division, the first meiotic division, which also kicks on then ovulation. So what happens is, this oocyte gets kicked out, and what gets kicked out in ovulation is now the secondary oocyte. And what's left on the ovary are the thecan granulosa cells that differentiate into a gland called the corpus luteum. So the corpus luteum is this area up here of granulosa and theca cells that kind of stay in the ovary. And the corpus luteum is very important because it makes estrogen plus progesterone, which changes the hormonal profile in a very significant way. Pardon? What is that? I will get to that. That's an advertisement for the next topic. Okay? Any questions on this point? So what's going on in the ovary is a lot of really important stuff. First thing is you got proliferation. A follicle wins out, you get a lot of proliferation. You get lots of granulosa and theca cells. The difference hormonally between prior to the proliferation and after the proliferation is huge because it changes the nature of the feedback system. And then after ovulation, you get the corpus luteum, and you get estrogen plus progesterone, which also plays a big role in the menstrual cycle. And this is where it's all coming from. So any questions? Because I'm going to put this together, what we just talked about in a graph that should pull this together. Yes? Yes, the corpus luteum comes from the granulosa and theca cells that are right at the site of ovulation. So granulosa cells plus theca cells differentiate into the corpus luteum at the time of ovulation. So there's a lot of changes taking place. Fluid pressure buildup, a surge in LH, meiotic division, basically metabolic chaos, and then you get the secondary oocyte getting produced. Yeah, that's correct. When does the question is, when does the differentiation take place? It begins right at the time of ovulation, because that's when you want estrogen plus progesterone to be produced. And there's a reason for that. And you'll see that in a matter of 2 or 3 minutes, maybe 4. Okay? Other questions? Yes. Your name, please? Okay. Yeah, the question is, does it stimulate release? Let me take that a little stronger statement. The corpus luteum produces and synthesizes estrogen plus progesterone. Because remember, it comes from the theca cells and the granulosa cells. Both of them are steroidogenic cells. One can make testosterone, the other can make estrogen. Well, it turns out progesterone is a sort of a compound in the pathway of making testosterone. So they're already steroidogenic. So you have 2 types of steroidogenic cells come together, and you get a gland called the corpus luteum, which then makes estrogen plus progesterone. It's very important to see this, because hormonally these cause very big differerences. Yes? No, this is the first meiotic division. Oh, yeah, good point. Thank you. There's a little polar body. That's Danny DeVito. He just kind of, versus Schwarzenegger. Okay? Yeah, you get rid of that one. The other polar body, by the way, hangs around in the first cleavage states. Hangs around there for a little bit. That's the second one, the haploid one. This is not a, this is 23-2C. Right? Yeah, I don't consider a true haploid because of the DNA level. Both of these are 23-2C. Okay? Other questions? Very important to see this. All right, so let's pull this all together in the menstrual cycle. And I don't think we need this part anymore. So the menstrual cycle really is directed by, all these hormonal changes. So first let's look at the steroids, the steroid hormones that are produced. Okay? So here's ovulation. And ovulation is about halfway point, so this is about approximately 14 days. It can vary from person to person. The second part after ovulation is about 14 days. So I'll put this down here. Here's the steroids. This first part is called the follicular phase, which is follicle development. So the first 14 days, you get all this stuff, follicle development. In the second 14 days, it's called the luteal phase. These are the phases of this. And that's because you have the corpus luteum around. Remember, we only see the corpus luteum after ovulation. So let's bring this down. So estrogen starts out low, and because of proliferation, you see the point between the kind of immature follicle and the much more developed follicle, you have a lot of proliferation. So as a result of the proliferation, you start increasing the level of estrogen. And then it stays up, it comes down a little bit, and kind of stays up. This is all pretty high. When do you see progesterone in the menstrual cycle? Only after ovulation, right? Because that's the only time you see the corpus luteum is after ovulation. So progesterone then comes blazing high. So the 3 hormonal states is low estrogen, high estrogen, and estrogen plus progesterone. And these define 3 distinct regulatory aspects of the menstrual cycle. When you go from low to high estrogen, due to the proliferation of the granulosa theca cells and maturation of the follicle, what you call low and what you call high varies for each individual. There's not a definable line that everybody crosses. It's different for every person. This threshold is different for every person. You could probably say across 1 million women, you could probably have an average what this really means. It seems arbitrary, but hormonally, what it does, the ramifications, when we talk about the feedback system in a second, is completely different. Okay? Any questions? You go from low estrogen to high estrogen to estrogen plus progesterone, dictating 3 distinct regulatory phenomena in this system. Okay? All right. Oops. Now, one of the ramifications of this increasing estrogen is a surge in LH. So let's just continue the ovulation, the stud line's ovulation. So let's look at the anterior pituitary hormones. Okay? LH is pretty low, pretty constant. Also, there's a big spike in LH. And the same thing, there's less of a spike in FSH. But they both spike a little bit. And then it comes back low. So you get this surge, this LH surge, just prior to ovulation because it's one of the signals of ovulation. And as you'll see, the surge in LH is kicked on by high levels of estrogen. Not kicked on by low levels of estrogen, and absolutely not kicked on by estrogen plus progesterone. So in this profile, we have high levels of estrogen. That's where you get the surge in LH. Does everybody see that? That's a very important point to see here. Then the other part of this is in the endometrial lining of the uterus. If we just look at thickness, first couple days of the menstrual cycle is the menstrual flow. And then it builds up. Here's ovulation down through here. Builds up and builds up. The optimal thickness for implantation of the blastocyst, which we will talk about at some point here, is about 7 to 8 days after ovulation. So what does that tell you? After fertilization, it takes about 7 to 8 days before anything that can implant is in the vicinity of the uterus. It takes 7 to 8 days. And that's why the system is timed, so that the optimal thickness is, in fact, 7 to 8 days after ovulation. Questions on that? It's very important to see the timing aspect. I'll leave this on the board when we talk about the hormones.